Welcome, everyone. Welcome to the Halophila Stipulacea webinar. Before going to the webinar and moving forward, we'd like to have a dedication to Susan Williams. Susan was tragically killed in an automobile accident this last year, on 25 April, and we'd now like to take a couple of moments of silence in her behalf. With funds provided by Puerto Rico Sea Grant, Virgin Islands Coastal Zone Management, VIEP score, and the University of the Virgin Islands, and the Center for Marine and Environmental Studies, University of the Virgin Islands, we now present this webinar focused on the seagrass Halophila stipulaceae, a non-native species in Eastern Caribbean waters. The webinar is structured around 5 15-minute presentations. The list of speakers and topics appears on your screen. The first two discuss general characteristics about the species and its relatively rapid spread. The next two describe results from recent studies in the U.S. Virgin Islands, and in the final presentation, potential management options are discussed. Following the presentations, we will have 45 minutes of recorded discussion. Antonio Farchetti will now provide a few ground rules for this session.
Good afternoon, everyone. I'm Antonio Farchetti. Thank you once again for joining the Halophila stipulacea webinar. So in order for everyone to hear the presentations, we will have everyone muted during the webinar. We ask that you hold all questions to the end, which will be answered during the discussion portion. We will be making this webinar available as a recording for future viewing online. I believe first we will be having Paul Jobsis.
All right, good afternoon or good day, everyone. This is Paul Jobsis, and I'd like to go ahead and get started. Apparently, we have a little bit of a technical problem. Brigitta is in a different webinar, so we're trying to get her into this one. I will go ahead and proceed. We're talking about the impacts of Halophila stipulacea in the Caribbean seagrass communities. Picture there is of 3 native, Halophila, Seagrass testudium, and Stanford filiforme, and of course our honored here, Halophila stipulacea. Most of the work that I'll be talking about was conducted around the U.S. Virgin Islands, but I will also be using some information from Marjolaine Christensen, whose excellent paper I've been using a lot recently. So I see she's online. So, okay. All right, so what are the observed effects of Halophila stipulacea in the Caribbean? We've seen some dramatic changes in seagrass composition resulting from Halophila stipulacea becoming dominant species in the area, altered recovery of seagrass beds following disruptions, and we've also seen the altered grazing by mega herbivores, invertebrates, altered seagrass communities as well. So this data is from St. Thomas, Brewers Bay, U.S. Virgin Islands, and it was collected by Tyler Smith and his students over the past 10 years. It shows a dramatic change in the seagrass in Brewers Bay. This is 3 areas within the bay, approximately 100 samples per year. So we have you can see in 2009, it was predominantly Serengodium, filiforme, a little bit of Thalassia, and a little bit of Halophila. 2010, we had Hurricane Earl come through here. It was actually a tropical storm which turned into a hurricane just as it passed over St. Thomas, caused a lot of disruption into Brewers Bay, including wiping out most of the seagrass, and also most of the sea turtles left the bay. 2011, the level of seagrass were still very low, and it slowly started coming back 2012, 2013, 2014. As you can see, in 2014, we get the introduction of Halophila stipulaceae. And Halophila stipulaceae was actually first observed in Brewers Bay in 2013, but it becomes a dominant species by 2015 and goes over, again, 2016, becoming over 95% of seagrass that was observed in the bay. 2017, of course, we had Hurricane Irma, which caused much more disruption into Brewers Bay than Hurricane Earl. However, immediately following the storm, that October, they were able to measure seagrass in the bay, and there was still a good bit of Halophila in the bay, and that has done nothing but increase. We'll point out that there is some Syringodium. You can actually see it at the very bottom of that 2018 bar, but is dominated by Halophila now. This is similar to what's seen, I guess, in Lac Bay from Christian's paper. What they're showing here is in 2011, they did have Halophila in the bay, but most of it was Thalassia, a little bit of Syringodium. By 2017, Halophila has increased and basically pushed this Thalassia into shallower waters, or it's being replaced in deeper waters, and believed by turtles grazing and it moving into the grazed areas when the turtles graze the Thalassia in shallower and shallower areas. So this is what they found in Lac Bay, and again in the non-grazed areas. If you look at graph A here, the white circles are non-grazed areas. You see very little change in the Halophila, whereas where turtles are grazing, it increases, and again that is represented by the decrease in the Thalassia. Okay, again, some more observations from Christensen, and then we also will talk about Brewers Bay. They did a nicely designed cafeteria study, and this is directly from their paper, and showing again that the turtles, 30 turtles were observed, and again they were increasing they were choosing the Thalassia over the other two seagrasses. On the right in C and D, we're showing the green sea turtle in Brewers Bay and a typical, what we would call a mixed patch of Halophila and Syringodium. So the turtle there looks like it may be feeding on Halophila, but again, you see the Syringodium in coming up through that, and we've had many examples where we're observing there. They usually will come out and just pick the the Syringodium out of the Halophila. Of course, there are other grazers as well. In the bottom right corner, you see there's an artificial reef that was used for a study in a class. We had, there are 12 diadema sea urchins around this, and they're creating basically a halo. When they are feeding on the Halophila, that's basically Halophila all the way around it, and keeping that area clean and then going back to the reef, the artificial reef. So one of my students, John Cassell, put in a lot of work looking at sea turtles and their use of different areas in the bay, whether it was related to native seagrasses or the Halophila. And so one of the methods they used were swimming belt transects. So the photo there is a photo of them doing one of their transects along the bay, and John's there sort of in the middle, and he is basically recording when somebody sees a turtle, green sea turtles, whether they are over native seagrass, mixed seagrass, invasive seagrass, or sand. He then normalizes data for the amount of Halophila. This was in 2016 and early 2017, so we're definitely getting dominated by the Halophila, but yet we see when you normalize for the area, the relative area of the seagrasses, the native seagrass and the mixed seagrass still has much more turtle density, so to speak, than the invasive seagrass. Okay. John also did some behavioral studies, so that's John on the upper left, he followed 30 turtles. I should say, if I can go back real quick, this was 45 weekly adult transects and 513 turtles were observed over that time. It was in the same bay over over a 1 year period. Okay. Also in this bay, John did some behavioral studies where basically he floated us him on the left, a big black arrow is pointing to a turtle, which he's observing, and he basically watched 30 turtles for 30 minutes and wrote down what they were doing, as well as having a little GPS thing on his board there, and he could track where they were going through the bay. And basically what he's found out is they spent most of their time grazing. That's not a big shock to anybody who's seen sea turtles. But again, they were grazing mostly in this native and mixed beds. And he actually never saw them during these 30 samples graze in a monotypic Halophila bed. We have seen that elsewhere, but not during these 30 samples. Okay. John also worked on some mesoconcym studies looking at diadema and whether he gave them a choice or cafeteria type of experiment, again with Halophila, Syringodium, and Thalassia. And he let these the sea urchins in the 24 hours. They were not starved or anything like that. They were just captured, put into the tanks. He had, of the 70 that he trials, 48 ate some of the grasses, and again with significantly eating more of the Syringodium than of the other two species. Again, Halophila and Thalassia were not very different. Okay. Let's switch over to looking at juvenile fish communities. This is a paper by a former student, Lauren Ollinger, and her cohort on what they did is they put small fish traps, basically catching juvenile fish. They put 4 fish traps into 3 different bays and repeated this 5 times. They would allow them to soak for 24 hours. They'd come out and count and measure the fish that were in there. And so these letters down the bottom, F, L, and S, are for Frenchman's Bay, Lancashire Bay, and Sprat Bay. So they aren't significant other than that. They're just 3 bays. But what they found was interesting. They found that in the Halophila stipulacea, many more nocturnal carnivores, carnivorous fish, and very few of the herbivores or the diurnal carnivores. So it made us really think that this was something worth repeating. So we tried that again for another study. This is just within Brewers Bay. They set out 5 traps for 12 hours, and they separated this in the night and day. Since they saw this difference in the night fish that they would catch, nighttime predators versus daytime predators, they wanted to work on that question some more. So 5 traps set into the bay for 12 hours, so 12 hours a day, 12 hours a night, 5 times, and repeated 5 times, so 25 samples per day and 25 per night. They found very similar results here. Again, they actually found very similar number of fish would come into the traps, especially at night. Daytime was a little bit less in the Halophila patches. They found the nocturnal carnivores were much greater in the Halophila traps, whereas the diurnal carnivores and the herbivores were found much more in the Syringodium. The reason they did not do Thalassia is because Brewers Bay has very, very small patches of Thalassia. Okay, so what their conclusion was that the sort of reduced family diversity and altered juvenile fish assemblages in Halophila stipulacea were driven by the abundance of some nocturnal carnivores and the scarcity of herbivores and diurnal carnivores in the Halophila patches. Okay, so native herbivores appear to prefer the native seagrasses. That's kind of my question was why. And again, Chris Christianen's paper did a great job of looking at the nutrient levels, found that they had basically the Halophila stipulacea is an inferior food source with lower soluble sugars, lower percent nitrogen, phosphates, and than the native seagrasses. John Cassell did a very similar analysis, but he did not find any difference in the nitrogen or phosphate levels. He did find, he did not do soluble sugars, but he also found lower phenols and lower acid digested fibers were in the Halophila stipulacea, which should make them more palatable for animals. So whether this is just some type of behavioral response that they're not used to eating this, we know that they can eat it. They do. Green turtles and of course the diademas have been recorded eating it. So not sure why, but something definitely is happening. I'm going back to this slide here showing the change in the seagrass community here in Brewers Bay. Again, showing this really as a dramatic shift that we've been seeing. We are seeing overall more seagrasses, higher percentage of the benthic is covered with seagrass, including going down to close to 90 feet in Brewers Bay. We still get Halophila growing that deep. We also, it's interesting to look at the difference in the recovery after these two storms. With Halophila there, it came back much quicker and again removed more of the native seagrasses. So this is a real issue that's going to have to be addressed, not just here in the Virgin Islands or in Bonaire, but throughout the Caribbean. And because these seagrass beds are changing quite dramatically, Brewers Bay is no exception. There are similar bays throughout St. Thomas, St. John, and the Caribbean that are being affected by this Halophila. So again, just to repeat some of this, we found that Halophilas in a wider range, depths and salinities, and we didn't find that, others have. Recovers more quickly following disturbances. There's perhaps an enemy release of native grazers, which benefits Halophila stipulacea, as they basically are feeding on native seagrasses. Altered seagrass fish communities with reduced herbivores are found in these Halophila seabeds, and they're also able to disperse over a wide area by fragmentation and drifting. So I want to thank you for listening, and we're going to save our questions for the end, but I need to thank especially the Association of Marine Labs of the Caribbean, who supported John, VIF Score, who supported me, as well as Antonio here, our local coastal zoning and Department of Planning and Natural Resources, the UVI Sea Turtle Club, and of course, the University of the Virgin Islands. All right, thank you.
All right, thank you very much, Paul. So next we will have Brigitta van Tussenbroek present.
Good afternoon, everybody. I'm sorry I was late. We had the wrong direction for this Zoom meeting. Can you hear me? Okay, then I will continue. So I will briefly describe the botany and life cycle characteristics of Halophila and Stipulacea in general. Halophila stipulacea is a well-recognized species. There are no taxonomical doubts about its status, and it was first described in 1867. There are about 9 synonyms of Halophila, with the genus varying between Barkania, Zostera, and Thalassia, and the species denominator varying between Bullata, Stipulacea, Balfouri, and Madagascar cascariensis. It belongs to a very interesting family, Hydrochaetaceae, which is a very large family, mostly consists of freshwater species. It has a large variety of reproductive strategies. So the plants can either be monoecious, dioecious, and flowers can be perfect or unisexual. The flora nectaries can be present or absent. So also pollination in the species of this family can either be biotic by animals like insects on land, entomophilus, or it can be abiotic by water, which is the case for most seagrasses. Although we recently found that Thalassia testurinum can also be pollinated by marine fauna. Within this really very large family of Hydrochaetaceae, the seagrasses form a monophyletic group. And they're based by the genus, they're formed by the genera Halophila, Thalassia, and Halus. So Halophila actually is the most diverse genus within all the seagrasses, the marine angiosperms. All Halophilas are small and fast growing, a large phenotypic plasticity, and a high tolerance for temperature, salinity, or water depth. The subdivision is this genus is based on vegetative characteristics and basically on the organization of the shoots and the position of the flowers. There are some species within this genus which are taxonomically not really, there's some taxonomic doubt, for example, the whole Halophila ovalis clade, which you see on the top. But the other species within this genus are really very well defined and there are no doubts about them. These plants can either be monoecious or dioecious. They're all pollinated by water. And when they are sexually reproductive, they form small fruits, which usually have a moderate number of seeds. And the sexual reproduction can actually be incredibly high. For example, Halophila decipiens can form almost 10,000 seeds per square meter. And these seeds are very small and have a hard seed coat, which are capable of dormancy, which is not always the case for seagrasses. The clonal growth. The clonal growth in seagrasses depends on the rate of the rhizome elongation, the distance between the nodes, and the way they occupy space depends on their rates of branching and also the angle of branching. And all Halophila species have actually a meristem or a dormant meristem at the base of each shoot. So actually their branching capacity is very high, which allows them to occupy space very efficiently. What do we know about the clonal growth rates of Halophila stipulacea? Not very much, actually. At least what I found in the literature, Babe in the Gulf of Aqaba found that shoots are formed with a mean rate of 1 shoot every 8 days approximately. And the lifespan of the shoots or the leaves is quite high, 74 days. About the non-native Halophila stipulacea, Willette and Ambrose found that beds, actually, so they actually metal a patch, expands about ½ centimeter a day on average, but can expand at a maximum rate of 6 centimeters a day. And Smulders et al. In Lac Bay, Bonaire, they studied actually the clonal fragments, the settlement, and they found that a fragment can settle within 10 days at a success rate of 100%. And these settled fragments actually grow very fast, on average almost a shoot a day, which is a lot faster than the 1 shoot in every 8 days registered by Wabe in the Gulf of Aqaba. But we don't know much about clonal growth rates of Halophila stipulacea yet. What we do know is generally in seagrasses, there's a very close relationship between the rhizome thickness and the clonal expansion rates. For example, this graph adopted from Maban Duarte in 1998, you can see that when the rhizome diameter differs, for example, a patch can grow, so a shoot or a small fragment can grow into a patch of 1 meter in almost a year, in 10 years, or almost 100 years, in the case of Posidonia oceanica, which has really very thick rhizomes. So Halophila stipulacea is more in the range of the green one of Halophila ovalis because it has an average diameter of 1.1 millimeters. About the sexual reproduction, Halophila stipulacea is dioecious. And these are pictures which were given by me by Juan Bin Yongyen. I hope I pronounce his name right. And you can see it has separate male plants and separate female plants, and the fruits produce quite a lot of really small seeds. Actually, dioecious means something for an introduced species, because if a species is introduced in a new area, the introduction must have been taken place at least 2 independent events, because first arrives a male, the next one actually 50% chance it's again a male, and then 50% chance again whether it's a female. So if a non-native species and dioecious species establishes successfully in a new area, you can almost assume it mainly occurs through clonal reproduction. What do we know about the dispersal capacity of the sexual reproduction? Not much, again, of this species. I found in the literature that Halophila stipulacea has, as most seagrass, a dual dispersal mode. So the reproduction of the fruits can either open in situ, liberate the seeds, and aid in the general genetic diversity of the population itself, or the flutes or fragment can float to the surface and disperse by the currents and wind over larger distances. Of course, how far depends on the floating time, and which is very short for Halophila stipulacea. I found for Malm in 2006, it's only a couple of hours. And what you can see here, for example, in the Caribbean for Thalassia testurinum, on the left, there's a coastal transport, and that's a floating time of a couple of hours to 3 to 4 days. And the dispersal capacity is quite low. It's just mainly local. And there's only significant long distance dispersal, so over large distances, when the fruits can float at least a couple of days, if you can see on the right. So actually the long distance dispersal capacity of reproductive, of products of the sexual reproduction is rather low for the species as far as we know now. The flower phenology, the Red Sea versus the Mediterranean, where the species was introduced a long time ago. For example, in the Red Sea, it's mainly female dominated, whereas in the Mediterranean, in the Eastern Basin, it can be either male, female, or more or less equilibrium between the two. And the reproductive season is more or less similar in the Red Sea, where it's native, as in the Mediterranean. Maybe the Mediterranean a little bit longer. So what would he know about Halophila stipulacea and its reproduction in the Mediterranean? Well, it was introduced through the Suez Canal in the late 19th century. And actually these drifting fragments already were observed with female flowers and young fruits. And 30 years later, they only found female plants in the Eastern Basin. But again, in the half of the previous century, they found plants of both male and female plant parts, suggesting that new fragments of another sex had been introduced in the area. And afterwards, they just find intermittently sterile and fertile populations throughout. Halophila stipulacea in the Mediterranean was restricted to the Eastern Basin for almost 80 years before it spread further out into the Mediterranean. And it was also only found in the middle of the Mediterranean until 1970. And in Italy, it has been reported since 1990. And in Italy, they have only found male flowers until date. But the population has very high genetic variability, which was determined by rapids, by Procaccini et al. And they also found a sort of genetic divide in the Halophila population, which was mediated by a depth gradient. So they concluded that, and Procaccini and Gambi et al. Concluded that the main propagation of the species in Italy is by fragmentation and clonal growth, but the high genetic variability in Italy suggests multiple settlements by genetically different individuals. What do we know about flowering in the Caribbean? Well, Halophila stipulacea was first recorded in Flamingo Bay in Grenada. In 2002, it was vegetative. In June 2014, the first male flower was recorded in Venezuela at Playa Mansa. And in April 2017, the first fruits were detected in the US Virgin Islands by Chiquilo et al., which is just recently been published. But they haven't found any seeds until date. And again, it's a dioecious species. So what does this lack of seed development, it has been found before, for example, in a non-native Halophila ovalis in Antigua, also the Caribbean, no males, but they did find fruits actually. And within the fruits, they found ovules. But these ovules never developed into seeds. And the same, for example, has been found for Halophila johnsoni, which only has female flowers also in Florida, no males. And Yorick et al., they studied the embryology of this species, at least the development, the genetic development of this species, and they found a normal gametogenesis. So, and a normal embryo sac was formed with a haploid egg cell within these ovules. So this means that the lack of seeds as the Halophila ovalis, the Halophila johnsoni population, is not because of some sort of abnormal reproductive development. It basically means there's no pollination occurring. And also that obviously there's no apomixis occurring. So what can we do about future research of life cycle, in terms of life cycle of Halophila stipulacea? It would be very interesting to determine actually clonal expansion rates based on models of clonal growth. So measure branching angle, branching rate in different environments. Because we don't really know how fast this species can spread under different conditions. Sometimes the species is just a noble undergrowth, and sometimes it really acquires invasive characteristics, completely removing all the native species. So it would be very interesting to study how fast this can go under different scenarios. It's also very interesting to study whether viable seeds are being produced, which can be either through studies of development, megagametogenesis and embryogenesis, or study seedling development. Also, the capacity for dispersal and long distance dispersal would be very interesting to study, which can be by fragments, which has been done a little bit by Smulders et al. Already, and can be happen in season floating fruits. It will be interesting to see if floating fruits can float for longer time than a couple of hours. And of course, another way is to study population genetics and see if the population we have in the Caribbean is genetically different, and if it's different, whether the genetic differentiation increases with distance, which gives an idea about the dispersal potential. These are the aspects I would like to cover of the botany and life cycle characteristics of Halophila stipulacea, and thank you very much. And again, the questions will be at the end of this seminar.
Thank you, Brigitta. Next, we have Demian Willette. Can you do it?
All right, can you hear me?
Yes,
Okay,
not
wonderful.
very clearly.
Not very clearly. Let me attempt this. You hear me now? Better?
Yes, a lot better.
Okay, wonderful. All right, thank you all for participating in this webinar. Again, my name is Demian Willette, and today I will focus on the spread of Halophila stipulaceae in the Caribbean. All right, give me one moment, I apologize. It looks like this box is covering part of my screen. Can you see the screen clearly? I apologize. Okay, perfect. Okay, oh, that's not any better.
Damian, we can see your slide.
You can see my slides. Okay, on my screen they're partly covered, but no problem. Okay, so I apologize. So there are numerous examples, of course, of invasive species in the world's marine environments. We can see up on the screen here, there's some examples, Sargassum, lionfish, and the green mussel. And we're using the term invasive to be defined as a species that's introduced outside of its native range, and then typically it's associated with some ecological or economic consequences. Given that, there are rarely have there been examples of invasives in seagrasses. An example of this, however, is Zostera japonica along the Pacific Northwest coast of the United States. You can see the picture here at the top left. In the foreground, we have Zostera japonica, the dwarf eelgrass, and then in the background of the photograph, Zostera marina, regular eelgrass, I guess. And so this species occurs at a number of locations along the Pacific Northwest. And for the most part, it's believed that the two species don't interact. Now, there is a recent study that suggests when the two are grown in similar conditions, such as increased level of total carbon dioxide, it is actually the invasive or Zostera japonica that performs better. So at this point, we don't see those examples, but the spread of Zostera japonica to this region may change over time in terms of how it interacts with other native seagrasses. The other example of an invasive seagrass would be Halophila stipulaceae. I like to call it the world's first globally invasive marine angiosperm, a beautiful plant, I think, pictured right here. It is native to the Red Sea and the Indian Ocean. It is a salt and light tolerant species. Let me put that briefly. So in terms of light, I've observed it growing as shallow as less than 1 meter of water depth. You can see an image on the right here that says leaf reddening. These are anthocyanins that the leaf produces under high irradiation, as I understand. But additionally, I've seen it down more than 18 meters depth. So pretty tolerant in that sense. Fast growing, top heavy seagrass. All of its biomass, the majority of its biomass is above the sediment. However, in its native range, it is a key primary producer of marine ecosystems, feeding things such as sea turtles. So how has this species spread around the globe to be our global invasive? Well, again, indigenous to the Indian Ocean and the Red Sea. In the mid-1800s, the opening of the Suez Canal spreading into the Mediterranean Sea. Over the next 100 years, spreading across the Mediterranean, as Brigitta just discussed, making it as far as Italy and Sicily. And then at some point, it spread across the Atlantic, being described in the Southern Caribbean in 2002. Okay. Additionally, we have some interesting data that I'm happy to chat about during Q&A that suggests maybe there was another route for the seagrass to make it to the Caribbean based on some niche modeling. But let's talk about the Caribbean specifically. So at the south or at the bottom of this image, you see South America continent. To the north would be heading towards the U.S. mainland. And in 2002, we had the seagrass arrive in Grenada, or at least first described there. The size of the circles are giving you an indication of the coverage of the seagrass in hectares. So a very small amount in 2002. I found it on the island of Dominica in 2007. And then in 2008 on St. Lucia. 2010, we see more places where it's being spread, or at least where it's being found, described, including Bonaire, St. Vincent in 2011. 2012, we see it in a number of places, including its northern limit of St. John and the U.S. Virgin Islands. In 2013, more locations. Importantly, in 2014, it makes it to the mainland of South America and Venezuela. And then more recently to Puerto Rico in Culibre. All right, so we see that the seagrass has spread across the Caribbean. How is it doing that? Okay, so I want to address this question: is Halophila stipulaceae something to worry about in the Caribbean in the scope of this presentation on spread? Well, it spreads both naturally and artificially. Okay, fragments of Halophila stipulaceae are created by storms that rip the seagrass up from the substrate and liberate it from the bottom, but also by activities such as anchoring, pictured on the left here, There you have a metal anchor tearing through the middle of the seagrass bed, and this is an observation I made where then the fragments were freed, as well as things such as fishing, and this is a West Indies fishing trap that also the seagrass grows into. It's removed, those fragments are liberated. The fragments are viable even at quite small sizes. Okay, so we did a couple of experiments where we created small fragments as small as 1 centimeter, actually, 1, 2, 5, and 10 centimeters, and we allowed them to float in the water column. And what we found was as small as 2 centimeters, they can survive and subsequently settle. And what we found was that over 7 days, all of the fragments that were at least 2 centimeters in length, including 1 vertical shoot, increased up to about 23% in total biomass and an increase in 20% of length. Okay, so pretty viable in that sense, so pretty small pieces too. Once they've settled, how do they perform? Well, the lateral growth can be quick, and Brigitta had gone over this. I just want to reiterate it a bit. The rapid lateral expansion has been measured at 1 centimeter per day with a maximum of 6 centimeters. That was measured in Dominica. We've measured the 1 centimeter of expansion per day on multiple islands in the Caribbean. And for comparison's sake, this is like Clonal taxifolia. Here we have it in California. We call it killer algal because it can cause some problems locally here, but that's pretty fast comparing an angiosperm versus an algal species. Additionally, it grows in all sorts of habitats, bare sand, in pure sand being just by itself, and in mixed beds with native seagrasses. And so on the bottom left, you can see just there's some Halophila stipulacea growing by itself. On the right, the photo is showing it mixed in with Serengodium, those green spaghetti strings extending upward into the water column. And in some cases, it grows together in a really dramatic fashion. So this is a photo taken in 2017 from Princess Bay in St. John, Virgin Islands. This was a location that was dominated by Thalassia, as I was told, and then when we visited, we just found a few of these shoots of Thalassia and a lot of the invasive growing around it, kind of seemingly to choke it out a little bit. So what about that? What about this interaction with native seagrasses? So this is work by my graduate student, Kelsey, who was, I'll talk about a little bit more later. She did some mixed and individual mesocosm growth experiments. And what she found was that when Serengodium filiforme, native growing, it grew better alone, whereas the invasive grew significantly better when it was mixed with the native seagrass. We can see this in the box plots at the bottom. Okay, we can see the treatments, and strikingly, in that green box, there is the invasive seagrass growing alone. Next to it in pink, that large pink box is the invasive mixed with the native seagrass. So pretty striking there. What else? Well, we can look at these interactions with native seagrasses from field data. This is work that I did with my colleague Sasha Steiner, and in 2008 we swam around most of the island of Dominica and measured all of the seagrass. The total amount of seagrass was about 300 hectares, and that was dominated by Serengodium filiforme. You see the presence of other seagrasses. There's a little bit of Thalassia testanudum, a little Halophila radii, and in blue is Halophila decipiens. In dark green at the top, that's Halophila stipulaceae, just making up 19 hectares. We returned, and in 2013, we see a dramatic shift. Much more seagrass cover, as was mentioned earlier in the webinar. More seagrass is present, however, it's dominated by the invasive seagrass, and we see a shift in what natives are still available or present. Notably, Halophila decipiens, we did not see it again in Dominica, and we haven't seen it in Dominica since. Okay, so there's this elimination of one of the native seagrasses, namely a contour of the invasive. How else do we see that? Well, this is data from the U.S. Virgin Islands, work by my colleague Ranjit Krishnan and others, and Thomas Kelly of the Park Service. So we can look at data over multiple years. What you're seeing at the bottom, I apologize, it's a little bit blurry, but there you can see from 2000 to 2016, quadrat data measuring seagrass shoot count. under mooring buoys within the park. And what we see is that Halophila stipulacea increases after its arrival in 2011, yet we do still see the presence of native seagrasses. Some which we seem to be already on a slight decline before the arrival of Stipulaceae. We've done some work in terms of looking at this more quantitatively, and we see that there does appear to be an interaction between Stipulaceae and decipiens, where the native Halophila decipiens is being reduced by the invasive. So thinking about spread, what about the opposite of spread, being stopping or reversing it? And we say here, well, I want to show you just really quickly a figure that I developed. I want to emphasize it is hypothetical. Spread rate. What I've done here is that I've taken Grenada 2002 as the origin of this species in the Caribbean, and then plotted all of the first reports of the seagrass throughout the rest of the Caribbean. What that looks like is this. And so on the horizontal axis, you see year reported. That's the first year reported at a particular location. And on the vertical axis, distance from the earliest report, which is Grenada. If we use a linear regression to fit these values, we can estimate a tentative spread rate, which is approximately 37 kilometers per year. We can compare that to what was found for the species in the Mediterranean. Giorgio et al. In 2016 estimated the spread of seagrass was 12 kilometers a year using the Suez Canal as the origin. Okay, again, the caveats here are that we don't know if Grenada was the first place where it was introduced, and we don't know if there's been multiple introductions in the Caribbean. This just gives us an idea of, possibly how can it spread, or how quickly does it spread. If it arrives to a place, can we reverse that by removing it? Well, we've done this work recently within the national parks in the Virgin Islands. We removed 1 square meter plots of Halophila on scuba. Took an incredibly long amount of time, about an hour per square meter to get rid of every little bit. And then we monitored that for 14 weeks after, and within 14 weeks, invasive seagrass recovered by about 50%. And it also generated lots of fragments, which we're able to also reintroduce into the plots. The green line that you're seeing on this figure over time are the control plots, just to control for natural variation in the invasive. If we extend this out, we estimate that at about 7 months' time, the 2 plots will be identical again. So not perhaps the most effective use of removing the seagrass to the strategy. So in conclusion, 3 of them here. Halophila stipulacea had spread broadly across the Caribbean. It's on more than 20 islands since 2000. This includes seagrass beds, open sand, and coral reefs, and worth mentioning the sand halo that often borders coral reefs. Paul had mentioned that interesting data with the sea urchins. Where we've seen it, there's not that halo. So it occurs in lots of different habitats. It seems to spread primarily through fragmentation, but again, as Bridget just mentioned, Kelsey's paper that came out just in November confirmed the presence of fruits and flowers in the Caribbean, so it seems like there could be potential for sexual reproduction. And lastly, it's unclear if there are ecological or physiological barriers that may limit the spread of Halophila stipulacea in the Caribbean. Now, there's been some recent work looking at thermoclines and haloclines in the Mediterranean about the spread. We don't have that data yet for the Caribbean, but this is definitely worth seeing. Are there barriers to how this species spread, or can we anticipate it arriving to the Bahamas, to Florida, and Costa Rica around Central America in the coming years? That, I'd like to acknowledge my collaborators on this work, including the National Park Service and Coline, Thomas Kelly, and our funding source. And I want to thank you all for your time today in listening.
All right, thank you very much, Damian. Next, we will have Alice Stanford speaking.
Can you hear me?
That we can, Alice?
Can you see my screen?
Yes, we can.
Okay. Yeah, I don't know how to get rid of those boxes either, but hopefully they won't interfere. Good afternoon. I'm going to talk to you about a pilot project we did to determine the number of genotypes of Halophila stipulacea found around St. Thomas in the U.S. Virgin Islands. The background of my slide here is a Halophila stipulacea bed in Brewers Bay on St. Thomas. All right, not sure why my slides are not changing here.
Alex, I found clicking on the slide was the way to advance it.
Oh, there it goes. All right, so as has already been discussed, Halophila stipulacea was first found rooted in St. John in 2012, and then it was found rooted in St. Thomas in 2013. At the time when we collected our samples for this study, it had not yet been documented in St. Croix, but it was documented there later that same year in 2016. Since flowering is rare in the Caribbean, it has been assumed that the species spreads clonally. So we did want to know just how many genotypes of Halophila stipulacea were found around St. Thomas. We hypothesized that there would be multiple genotypes since it does spread quite easily. And we wanted to determine the genetic structure of the species as well as the clonal diversity in the area. So we collected samples from 6 bays on St. Thomas: Brewers, Lindbergh, Megan's, Lindquist, Sapphire, and Belongo, as well as Honeymoon Bay on Water Island and Lameshore Bay in St. John. At each bay, we laid out transects starting in what appeared to be the densest Halophila bed and ran the transects parallel to the shore, collecting one sample every 8 meters. And we collected 8 samples at each location, so 64 samples altogether. For each sample, we only used leaves that appeared healthy and were intact on the same rhizome attached to each other, so we could be sure they were all from the same genet. They were scraped with a sterile ruler in a sterile environment and rinsed with sterile water prior to DNA extraction. We tried a few different techniques, but we found the nucleospin kit to be the most effective for getting a good quality DNA sample. We wanted to get a large quantity of genome-wide data, so we used a RAD method, the 2B RAD method, as described by Wang, and we processed the data using methods and software provided by Eli Meyer. We were able to generate genome-wide SNP data. We didn't want to lose any of our samples since we only had 64, so we went with a relatively small number of SNPs for this type of data, which is 200. So that's still quite a large number compared to what you could get using other types of markers that are available. And we only took loci that had at least 10x coverage, so we wanted to be sure the data were accurate. Then we analyzed the SNP data using the GenClone software. This is just up here to get you a feel of what our data looked like. After it was processed, we generated a large matrix of data. Now, this is a small subset. It doesn't have 64 samples. It doesn't show all 200 loci. But from this matrix, we were able to assign each sample to a genotype. And if you look across the bottom of the slide, we gave each genotype a number. So on this slide, you can see there's a lot of genotype 1, a lot of different samples. The samples, each column is a sample. And then genotype 9, 10, 11 can be seen on this slide. And these are some of the loci shown in the rows. And those yellow highlights show you where there's a locus that differs from the predominant genotype of genotype 1. We calculated some basic population genetics indices just to get a feel for what's going on in the population. This PGen value tells you the probability of any given genotype being generated by chance through sexual reproduction. It's fairly low. It doesn't suggest there's much, if any, sexual reproduction going on. The p-sex value compares different individuals with the same genotype. So these are multilocus genotypes or haplotypes, if you like. And when they were identical, this was the mean probability that those were produced through sexual reproduction. It's extremely low, again, suggesting that those individuals we're referring to as genotype 1 are, in fact, all clones of each other. We did go ahead and calculate Ney's non-biased heterozygosity. That could be useful if we can compare to a data set that use the same marker sometime in the future, but it does suggest that there's some genetic variability in the population. And we also calculated the Shannon diversity for the genotypes. Again, extremely low number. If you were to go out into the water anywhere around St. Thomas and randomly grab a bit of Halophila, it would probably be genotype 1. That's almost everything is genotype 1 that we found. In the slide on the upper left, you can see the distribution of each genotype along each transect. And what you'll see is that genotype 1 was found on every transect, and all the other genotypes were just found from one of our samples. Now, our samples were collected fairly far apart, 8 meters apart, so there could well be more genotypes that we didn't detect, but we detected 16 genotypes from our studies, and Brewers Bay had 2 of them. Megan's Bay had 3. In Lindbergh, we found 6, which was the largest number of genotypes from any location. In Belongo Bay, we found 4, and also for Water Island. Lindquist only had 2, but in Sapphire and Lemishore and St. John, we only found genotype 1, just the 1. So the predominance of this genotype is something that was of interest to us. It was a little bit surprising to me that I found multiple genotypes, but most of them were found in very limited quantity, it would seem. This may just be an indicator that genotype 1 was the first to arrive in the area, and it just became established before the others arrived. It may be an indicator that genotype 1 is more successful in terms of competition. It would be nice to collect some more extensive data in order to test some of those hypotheses. Just to summarize, we didn't find very much genetic diversity, but we did find 16 different genotypes out of the 64 samples. Now, we think the most likely explanation for this is that there were multiple migrations of this species into the area. Certainly there are other possibilities. Sexual reproduction is possible, although considering that flowers are rarely seen and seeds have not yet been found, we think that's unlikely. The mutation or high mutation rate is another possibility. It would have to be an extremely high mutation rate to account for the amount of variation that we found. So again, we think that's unlikely. But it would be nice to get a bit more information to try and eliminate some of these hypotheses. We did go back in the summer of 2017 and do a more exhaustive sampling in Brewers Bay at 6 different transects where we collected a total of 105 samples, ranging from the low tide line out to 25 meters of depth. So these stars show the starting points of the transects. We've had some difficulties due to hurricanes and so on, but we have processed these data. They're ready to be analyzed, so we'll soon have some results on that. And also in the summer of 2017, Kyle Cheris, one of the members of our team, did map Halophila in the southwestern part of Brewers Bay. And what this shows you is in all the mapped area, except for this little purple bit, there was Halophila everywhere. And all this green is a bed of solid, pure Halophila. We had a lot of undergraduate students who worked on this project. I didn't write all their names on here, but I do thank them all. And I want to thank Kyle Jarris and Calvin Morton and Steve Retchford for helping us collect samples in the field. Also, I need to thank VIF SCORE, who was the primary source of our funding for this project. Oops, sorry. Here's our works cited, and I guess we're holding questions to the end.
Thank you very much, Alice. So this brings us to our final presenters, which will be Judd Kenworthy and Ashley Rufo.
Everybody can hear me, I hope. So I want to first of all thank Antonio and everyone who kind of stuck with the program here. I know it's been almost 2 years, and we had hoped to do this workshop in person, face to face. It would have been ideal, but I'm really glad that we're able to get it off the ground. So thanks a lot for all your hard work, everyone, especially Antonio and anyone who helped him down there. So what I'm going to do is kind of frame the management options for Halophila stipulacea in U.S. waters. And I'm going to start with a kind of an overview of what instruments and legal authorities would be available to us to consider in terms of conducting some form of formal management of the species. And then I'm going to turn the talk over to Ashley and Jocelyn, who work for NOAA, which is one of the federal agencies here in the United States that actually is kind of on the front line of working with the permitting process and all the issues that are related to the Magnuson-Stevens Act in essential fish habitat, which is really one of the principal drivers in our coastal management of seagrasses here in the United States. I'm not going to cover any international territories in my talk, but certainly that's something we might want to do in the near future, think about that. So forward here. So I've got 3 objectives. I'm going to provide you with some background and framework for the legal authorities that deal with invasive species in U.S. territorial waters. We already know we have it in the United States, in the Virgin Islands and Puerto Rico. It's also in at least one of our national parks, which is relevant to some of the federal regulations and plans that may be put in place to manage the species. And then I'm going to give you a brief overview of a case study and kind of suggest some of the lessons we've learned. As Demian showed you in his slides, we already have had some experience with a non-native introduction of a species, Zostra japonica, on the Pacific Northwest coast of the US, and it has also spread both north and south. And so we already have a management experience to work with, and we might be able to learn something from that. And then I'm going to turn the talk over to Jocelyn and to Ashley, and they're going to describe to you exactly what NOAA is doing right now with essential fish habitat. So in the United States, there are at least 21 federal laws that address non-native or invasive species. And you can probably, there's probably about 8 that would directly apply to a subaquatic species like Halophila stipulacea. One, of course, is the National Environmental Policy Act, which is, one of our, one that's implemented quite often in environmental regulation. The second one is the Magnuson-Stevens Act, which you'll hear about from Jocelyn and Ashley, and that is related to essential fish habitat and what role seagrasses play in that. This third one, the Endangered Species Act, will be kind of interesting. And given what we've seen today and what we know and what we've read about the species and how it's spread globally and how it's spreading here in the Western Hemisphere, I think it's safe to anticipate that at some point the species is going to enter the southeastern United States, perhaps in Florida. It may even get into the Gulf of Mexico. If it does enter Florida, it is going to be interacting with the Endangered Species Act because we have the only seagrass listed under the Endangered Species Act, Halophila johnsoni. And as many of you know, johnsoni and Stipulacea are very similar, and I think there's going to be probably a fair amount of interaction between those species. And you can go down the list here and find a number of different federal acts that could apply to Stipulacea. And then there's at least one federal executive order. This is a very famous order, 13112. I think it was 1992, somewhere in that range. This was the executive order that was put out at that time by the administration in charge of the US government that actually came up with a formal definition of what the federal government calls an invasive species. And that's an alien species whose introduction to us is likely to cause economic or environmental harm or harm to human health. This executive order has been applied to a lot of invasive species introduction. It's really famous for its application to issues related to ballast water. But these are a number of laws that could be applied in terms of developing any kind of management action relative to the federal government. We also have several federal agencies that share responsibilities, and they're kind of organized around these two councils or committees. There's a National Invasive Species Council, and there's an Aquatic Nuisance Species Task Force. And both of these are in place to develop and implement what is a national invasive species management plan. So believe it or not, there is a national plan. There is a plan that could be brought to bear if we wanted to, or if there was actually the impetus to do it. And that plan is designed to coordinate across federal, state, and local government agencies, to coordinate with private industry, other interest groups, and even private individuals in dealing with species introductions and invasive species. It has strategic goals that involve prevention, early detection, and rapid response, control and management, restoration, and formal organizational collaborations. One thing that this plan can do, and it has done a number of times, it can generate the kinds of support that are needed to deal with issues related to invasive species. And that brings in other government, non-government, state government organizations, staffing and funding opportunities, for example, with the National Science Foundation, with NOAA, other federal agencies. So there is a mechanism there to actually generate infrastructure, financial, and staff support to deal with some of these issues. Then we also have a whole mosaic of state laws. And I'm not going to go through all the state laws, but the bottom line is that we have a legal framework that is really a patchwork of federal and state laws, regulations, varying policies, within agencies, and many, many different programs. So there is this kind of dream of coordination and cooperation, but really what our experience has been in a lot of these issues, unless they're very, very high profile, like ballast water, it's a hodgepodge. So we end up with fundamentally different approaches to dealing with invasive species here in the United States. The case study we have with the seagrass is a perfect example of this. This is Zostera japonica. Demian mentioned it earlier. Its nickname is the dwarf eelgrass. It looks and acts a lot like eelgrass that we're all familiar with, but it's much smaller, and it does appear to have some unique characteristics that distinguish it from Zostera marina or the other subspecies of Zostera in the Pacific Northwest. And there's been considerable interest in the species. Demian showed you this slide earlier. We think it entered the Pacific Northwest sometime prior to 1957. It came on. People believe it came in with a shipment and importation of Japanese oysters. It spread north to British Columbia, and by 2002, it had reached all the way down into Humboldt Bay, California. So we had, sort of this interstate federal invasion, and also, these different states themselves. In the interim, in that period of time, some 50 years, a lot of research has been done. And I draw your attention to two review-like papers from the literature down at the bottom there, Schaefer et al. And Mock et al. These are very good papers that kind of summarize the science that's been done and also tie it to some of the management actions that have been taken. Really, if you read these papers, you look at the literature closely, you can really see that the scientific jury is really still deliberating about the species and what its impacts might be, and whether or not it fits in that, sort of puristic definition of an invasive species causing harm. Now, in the meantime, federal agencies have taken the position not to protect it. We're going to hear a little bit about that later on in the talk. Zostera japonica has not been listed on the federal invasive species list or the federal noxious weed list, yet the National Marine Fisheries Service in the Pacific Northwest that has chosen not to consider it in the same context as other seagrasses when it comes to regulations related to essential fish habitat. Well, the federal agencies have not acted, the state agencies have. The Washington State Noxious Weed Control Board in 2012 listed it as a Class C noxious weed. They said that Japanese eelgrass is non-native, difficult to control, and negatively impacts the shellfish industry. In 2013, this was modified to just simply say that Japanese eelgrass is now a Class C noxious weed. So what happened in Washington was the shellfish mariculture industry saw that this species was occupying a niche on unstructured mud flats. That was the ideal environment where shellfish mariculture was taking place. And they were worried that it was going to interfere. They were also worried that either the feds or the state might take a position to protect the species. So there was competition between the industry, the shellfish mariculture industry, and anyone who was interested in thinking about protecting the species. California also listed as an invasive and noxious weed. In fact, they seriously considered eradication. So in 2002, there was a cooperative effort between the University of California, Sea Grant, California Fish and Game, Humboldt State University, and they started working on what was called the Zostra Eradication Project. And they did a number of experiments using different techniques to find out if there were practical, cost-effective, and successful means to actually eradicate the species from Humboldt Bay to begin with. I suspect also with maybe plans for other parts of California. Now, in Washington State, they've since looked at using herbicides specifically directed at the shellfish mariculture industry to allow them to use herbicides to eradicate japonica in the shellfish farms. And I'm not really sure how far they've gone with this now, but you know, this whole concept of eradication is something that we need to have a really serious discussion about because of all the other environmental and ecological implications, as well as the probability of success. So what is the future for Stipulacea? Well, like I said, it's already in US territory, so there are mechanisms we could use to work on this species. There's quite a few of them. And the question of whether it'll end up in southeastern Florida, if that's the case, if it does happen, and it turns out that we react the same way we have with japonica, in all likelihood, the state of Florida, through the Fish and Wildlife Conservation Commission in their section on invasive species, will probably take the lead in this. And, you know, it's going to be, it could be some time, you know, it could be in the immediate future. But in the meantime, we ought to start thinking about how we might really plan appropriately what type of science we're going to use and how we're going to approach this if we actually want to think about managing the species at all. So right now, the front line of species management here for Stipulacea occurring in the Virgin Islands is being done by the National Marine Fisheries Service. So I'm going to turn it over now to Ashley and Jocelyn, and they can tell you about this program that's happening.
Good afternoon. Can everybody hear me?
I can.
Yes.
Okay, great. I'll assume everybody else can. So yes, good afternoon. My name is Ashley Rufo. I am a contractor for NOAA Fisheries, Southeast Region, and I work for the Habitat Conservation Division. The Habitat Conservation Division is responsible for implementing essential fish habitat provisions of the Magnuson-Stevens Fishery Conservation and Management Act. Our division has two different field offices here in the U.S. Caribbean that are dealing with Halophila stipulaceae on regulatory projects. So I am located at our field office on St. Croix, and I also work with Jocelyn Garcia, who is located in a field office in West Palm Beach, Florida, for the various projects here in the U.S. Virgin Islands. We do also have a field office over on San Juan, Puerto Rico, where we have our another federal employee, Jose Rivera, who works with essential fish habitat consultations over on Puerto Rico. Judd, next slide. Thank you. So the provisions of the Magnuson-Stevens Act mandate that the Caribbean Fishery Management Council designates essential fish habitat for various federally managed species. Essential fish habitat designations are intended to encourage the conservation of the habitat which federally managed species use for each life history stage. And as such, seagrass is designated by the Caribbean Council as essential fish habitat for species managed under our clean conch, reef fish, and also the spiny lobster fishery management plans. Now, although the council has not specified that only native seagrass is designated as essential fish habitat, I guess similar to the japonica situation in the Northwest, our consultations do not recommend any conservation measures for Halophila stipulaceae, the same as we would do for our native seagrass species. So essential fish habitat consultations are required with NOAA fisheries when federal agencies authorize, fund, or take an action that may result in an adverse effect to essential fish habitat. An adverse effect is defined as any impacts that could reduce the quality or quantity of essential fish habitat, either directly or indirectly. And through the consultation process, NOAA Fisheries provides recommendations for sequentially avoiding, minimizing, and then offsetting any adverse impacts through compensatory mitigation. Next slide. So for our current recommendations and best management practices, we include notifying National Marine Fisheries Service of any presence or absence in project areas, no relocation of any monospecific or mixed beds containing Halophila stipulaceae, preventing the spread from anchoring and bottom disturbing activities such as budding down in seagrass beds. We recommend removing fragments of Halophila stipulaceae that could be caught up in any turbidity barriers. And we also like to recommend to restore native seagrass areas so that they don't become over-colonized by Halophila stipulaceae first. And so far, these have been the actions that we have taken to help limit the spread as best as possible. Moving forward, we are looking to adaptive management. We would like to update and amend our current standing conservation recommendations and best management practices as new information becomes available, which is what piqued our interest and involvement in this workshop. So it's been great hearing all of this information about Halophila, and we will absolutely take it into consideration for modifying new recommendations and best management practices. And we are open to hearing any suggestions that anybody may have for mitigation. And that's all. Thank you.
Thank you both Judd and Ashley for that. This brings us to an end to our presentation segment. We will now be moving to a discussion portion.
So raise your hand.
All right, Judd, if you would please stop sharing your screen so I can move back to our PowerPoint.
You share yours.
Can you take it now?
That would be on your side. At the top of your screen, you should see a stop sharing button.
Stop sharing. I see new pause. Where's the stop? I got it. Sorry about that.
Okay, thank you everyone for joining us. So the discussion portion should last for approximately 45 minutes. You have two options for asking a question. The first is to raise your hand. In the participant panel, you should see a few buttons. One of them is the raise hand. By pressing that, you should see a little hand icon next to your name signifying that you wish to ask a question. We are also taking questions via the chat box. Either one will allow you to ask questions. And we have our first question here from Martina, who asks, hi everybody, I'd like to ask if the fragment viability of Halophila was tested in situ or in mesocosms? And this is to Demian Willette.
Okay, thanks, Brigitta. Our experiment, the data that I showed were in mesocosms. However, I think the work that was done in Lac Bay were field studies. So I believe there's evidence from two different studies.
Damian, would you mind repeating that?
Sure. So our work that was shown in the slides was in mesocosms. However, perhaps somebody else mentioned the work that was done in Lac Bay. I believe those were tethered fragments in the field. All right, I'll respond to that question by Abigail as well, if it's okay, Antonio. Why do you think Halophila stipulacea does better with native seagrasses than on its own? That's a great question. I am not sure. There may be a facultation of a seagrass already being somewhere in terms of settling, perhaps. I've done some reciprocal transplants of Serengodium and Halophila stipulacea in Dominica, and we just saw that the invade does well in terms of growing into them. I don't know mechanisms that might be leading to that.
Okay, we have a question from STCM Williams.
Hi,
right?
can you hear me?
Yes.
Yeah, this is Stacy Williams from Puerto Rico. We are presently mapping the distribution of this seagrass in Culebra Island, around Culebra Island in Puerto Rico. And it's more, my comment is more of a comment, not a question, but we've been noticing fragments floating along with sargassum. And so I'm wondering how this is affecting the float time of these fragments. And I guess, have a question is, has anyone looked into how sargassum is impacting, I guess, the spread of this seagrass?
If any of our presenters would like to answer that question.
Yeah. I'm unaware of anybody studying that.
I'm also unaware of anyone working on that. However, we have seen it tumbling together in sargassum mats. But yeah, I don't know if that is extending the dispersal potential or if it's helping it settle quicker into the sediment. That's a great question.
Yeah, let me think about it. I think it's a very good question. maybe we could just start testing it along as we go, because, for example, what really amazes me is that Halophila stipulacea has not spread a lot along the mainland of Latin America and South and Middle America, and it's mainly spread along the islands. If sargassum is a vehicle of transporting the fragments of Halophila stipulacea, I would really expect a really fast expansion along the mainland soon. So I think we don't know it right now, but maybe, unfortunately, we will discover it pretty fast in the next years.
Okay. We have another question from Hector Ruiz. I heard that Halophila stipulacea was collected at Dominican Republic. Can anybody confirm this?
Perhaps not.
I take that as a no.
Unfortunately, we don't know.
Okay. There's
Another question from Gary Rollerson. Hello out there. What are the defining visible characteristics to differentiate Halophila stipulacea from others, Halophila decipiens or Halophila anglemani?
Brigitta, can we ask you that question?
Well, the easiest thing is, I think Halophila stipulacea is the only Halophila species which has the basic scale almost completely covering the petiole of the leaf. So the other ones, you can still see the petiole sticking out, and Halophila stipulacea has the scale almost covering completely the petiole and part of the lower part of the leaf. And also the leaf tends to bend, isn't it, like a banana, which is easiest. And actually, somebody at my lab made a Seagrass ID application, which you can use on the Android phones, but until now it's in Spanish. So maybe, I hope we'll get an English version soon, and then you can just check it on your phone.
Okay, thank you. Our next question comes from Carolyn Pott. If you know you have an at-risk site, low seagrass density, sand, proximity to established beds, are there active management techniques one can use to prevent Halophila stipulacea establishment?
Yeah, you know that, Demian's study where they were removing that was in established beds already. I know in Brewers Bay, when we started seeing it come in, when we first identified it, it was a very small patch, probably less than 2 square meters. So it could have been dealt with at that point, perhaps, but there were more fragments drifting in, so it would take almost constant, upkeep.
Yes, I agree. I haven't seen things that can be actively done. We've attempted to work with fishermen in Dominica to clean their traps and so forth before redeploying, but I don't really know of things that could be done in the field to prevent at-risk sites. If I may, following up on the description of the seagrass, we do have a website that provides both visual photographs for monitoring as well as a way to report new sightings. I can, if you allow me to grab the screen for a minute, I can show that website. Just pop it up for a second and then take it back down.
Certainly. Demian?
Okay. Here it is. Sorry, I guess you can't see the full thing. It's very simple, invasiveseagrass.org. And so on the left, it has a bunch of things in terms of identifying the seagrass through metrics, but then also there's a report sighting function in English and Spanish, where then you submit it and we can track new occurrences throughout the Caribbean. And so this is funded in part through the National Park Service for the next 4 years. shouldn't stay active for that long. But this is one way if you want to see where we know it is so far, also where it's spreading to, but also to figure out is your seagrass thing you found really in Halophila stipulaceae. It also lets you submit photographs, which you can then confirm if it's that seagrass. All right, so I'll close this back down.
Hi, this is Judd. I wanted to ask about the genetics work, if possible. And is the genetics lab that you have going right now, is it, if in fact Stipulaceae were to arrive in the US continental waters, like in Florida or the Gulf of Mexico, are you capable of taking samples of that and doing the same type of work you've done to see if we could identify the genotype that we end up with?
Yes, we could do that. It just takes money to pay for the sequencing, pretty much, and the chemicals, but we have our laboratory is in good shape.
Mm-hmm. And so the other question I had was, do you have any plans in the future or funding to do any work where you might isolate these genotypes and culture them and look at some of their unique traits to try to better understand, for example, why you're seeing the predominance of genotype 1 versus the others?
We haven't thought about culturing them. What we had thought was it would be useful to monitor it repeatedly, to sample once a year or twice a year in the same location and see how the predominance of genotypes are changing over time. But also it would be nice to have a more exhaustive sampling, although we do have one for Brewers at least that's almost finished, just to get a better idea of how much of each genotype is out there. With 8 meters in between samples, there could well be larger patches of some of the other genotypes or additional genotypes we haven't detected. And that would also help us possibly think about how it's migrating, because it does appear that there are some areas that have fewer genotypes, that there might be something that's in some way protecting them.
Good, thank you.
Our next question comes from Rebecca Flynn. My question regards the fish community alterations. Is it possible that the overall size and shape of the Halophila stipulaceae blades provides less cover and is less preferred for hiding out in, and that nocturnal predators prefer it because it's easier to find a meal than in taller native grasses?
I'll answer that since I don't know if Lauren Ollinger is on the line, but I'm just going from her paper. They did spend a good bit of time measuring the different heights of the types of seagrasses, as well as that. So that certainly is possible. And so you're talking about 3 to 6 centimeter blade heights rather than the Phalacia or Sangodium, which might be 10 to 20 or so centimeters. So I think I see the next question as well. And Lauren, if you are on there, just raise your hand or start speaking up. And the next question looked like it was some also that I could take on. It was one about the BVIs, whether we are downstream from the BVIs, and we definitely are, but I don't think it's their fault because they're just downstream from St. Martin and all that. So it is coming with the currents and wherever those currents are coming from.
All right. Thank you. Our next question comes from Fee Smulders. Hi, Demian. Thank you for your talk. Do you know what time your fragments remained floating in the water column before landing on the sediment? And did they have quick shoot generation after settlement?
Okay, so thank you, Fee. We floated them in the mesocosms when they were recirculating tank at Veers down in St. John. They floated for 7 days, and at that point, only one of the, I think it's about 50 fragments had actually settled in that experiment. So, and I think it was about the 6th day or so that it finally did. So they were largely floating. Everything from, again, the 1 centimeter to 10 centimeter fragments stayed floating in the tank for that period of time. As far as did they have quick shoot generation after settlement, we ran another experiment where we same size fragments and kind of put them into the sediment and then pulled them out after a number of days and looked, and in all of those, in the vast majority of those, and if they were at least 2 centimeters in size, they started to show either new roots being put down or lateral growth of the rhizomes, even as little as a week. Now that fully warrants a larger study than what we did. Yeah, relatively quickly.
Okay, so we have another question. This is from Robert. Hi all, thanks for the informative talks today. I'm wondering, are there any ideas why Halophila stipulacea is able to outcompete native species such as Stipulacea testudum, since it is too short to outcompete the native species for light, and it doesn't lay down deep roots or rhizomes, so may not be outcompeting the native species below ground. Any ideas why it is effective?
Is Marjolaine online? She described it quite well in her paper. I saw her online earlier. Christiane?
Yeah, she's not.
Yep, you're online. If you want to unmute yourself, you can answer that question.
Hi everyone, can you hear me?
Yes, you can.
Yes.
Well, Robert, is this Robert Johnson asking the question?
This is from Robert.
Yes. Yes. He just visited the area where we studied the competition between the two species, Halophila stipulacea and Thalassia testudinum in Bonaire. And what we found is we did not, or Faye is currently studying the competition between the two species, but what we have studied already is the impact of turtle grazing on the competition, and that's what Robert has also seen in the field, I think. And then what we found is that turtle grazing selectively picks out the Thalassia, so making space for Halophila stipulacea to settle with fragments or to spread clonally, and then we found that Halophila stipulacea could spread faster than with grazing and without grazing. But if you don't have turtle grazing, we are also wondering why Thalassia, or why Halophila is outcompeting Thalassia. We also noticed a lot of building up of finer sediment at places where Halophila has settled, and yeah, we're looking into all kinds of aspects to study this further. So I'm not quite sure I didn't answer your question, but at least we know that turtles can impact the competition.
Yes, and we're seeing the same thing here with turtles. My student John Cassell also showed that the diadema sea urchins preferred the native seagrass, or at least preferred the Syringodium, and then you have this sort of what's described as sort of an enemy release form. So they are not being fed on by the native herbivores. The Halophila is not. So it has this protection while the native seagrasses are getting grazed upon. So it allows them a competitive advantage. That would be my suggestion as well. Although it's also well explained in Marjolaine's paper.
Thank you. Yeah, this is from Lauren Ollinger. Hi, Rebecca and all. Sorry I can't talk where I'm at, but yes, that is one reason that juvenile fish may prefer native seagrasses over Halophila stipulaceae. Other reasons may be Halophila stipulaceae provides suboptimal food resources, especially for herbivores, prevents access to the sediments underneath seagrass blades, especially for benthic foragers. These were just a few proposed reasons that juvenile fish assemblages may be so different between native seagrasses and Halophila stipulaceae. Judd, if you'd like to ask your question.
Yeah, I wanted to ask Demian a question about one of his slides. And I was fascinated by that one slide you showed, but you didn't really speak to it. Was the one where you drew the arrow from East Africa, the Indian Ocean, to the Caribbean as a possible route for Stipulaceae to get to the Caribbean. And so I had a couple of questions. One, of course, this relates to, what makes you think that possibility? Do you have any, sort of evidence or thinking about that? And is there any molecular data that might support that vector? And the reason I ask that is because that's one of the issues we've been dealing with Halophila johnsoni here in Florida, and that's, we do have some older molecular data suggest a relationship between johnsoni and ovalis, in that very same region of the Eastern Indian Ocean. So it would be interesting to, think about, these vectors and how these plants have gotten here, because we're still struggling with this ovalis johnsoni and whether or not johnsoni is actually, a unique species.
Yeah, thanks, Jen. And if you can give me the screen for a second, I will show where that idea comes from. So I work with colleague Elizabeth Rocco at University. Let me pull it up here. And so what we did here was, I apologize, it's the whole screen that you may be seeing, but it's a discriminant analysis based on environmental parameters based using known locations of the seagrass from around the world. And so the discriminant analysis you're seeing up on the screen right now, it shows different regions. Each dot represents a geographic location on the planet. And then how this kind of plays out is that it kind of groups like habitats together. And I apologize, I'm very quickly trying to find the figure for you, but we can draw a circle around the red Caribbean and the East India and the East Africa region. You can see kind of all clumped in the left portion of this slide. What that may suggest is that they share similar habitats. It's based on habitat features. Now that doesn't mean, it just says that those habitats are similar between where it's growing in the Caribbean, East Africa, and India. That is where we think, well, there's this similarity there. Additionally, we were digging into some human migration patterns and so forth, and this was very exploratory, but there was a big movement for individuals from India into the Caribbean and so forth, and there was a lot of boat traffic. This is really, kind of, stretching a little bit, but anyways, that would suggest that they are similar, at least the habitats they are currently existing in. More similar to the Caribbean, more similar to East Africa and India than it is to the Mediterranean or the Red Sea. That prompted a question that my student Kelsey Shaquilo is actually working on right now, part of her dissertation, along with Gideon Winters, who's on here as well, where we did SNP data for seagrass from around the world. We did the 2B RAD method that Alice described, and Kelsey's data was looking at the differences between the seagrass from its native region, the Mediterranean and the Caribbean. I don't have any slides I can share on that at this moment. Kelsey is writing it up right now, and we hope to have it out for review shortly. But yes, we have the genetic data. I'm just not prepared to talk about it at this moment. I apologize.
Well, that's okay. Keep me posted on.
Yeah, we
It'll-
hope to publish it
it'll-
soon.
Thank you.
We have another question by Caroline Pott. Is it unlikely that a 100% thick established Thalassia bed will be invaded without severe disturbance, hurricane, anchor scars? Were the changes in Lac Bay attributable to disturbance at all?
I think it's answered in the one below that.
And then Fee Smulders did respond, yes, invasions indeed. Unlikely without disturbance. The ungrazed 100% cover Thalassia border at Lac Bay has not been invaded by dense Halophila beds yet. The disturbance in the center in this bay has been caused by triple grazing. I'd like to open up to any of our presenters who wish to comment on that as well.
I would agree with the dynamics. Perfect sense. There just wouldn't be space for it to come in. Although we do get this mounding of the Halophila, which maybe could sort of overcome some of that, but there needs to be some disturbance. Whereas in the Syringodium, I don't think that's the case. It can just interleaf within the Syringodium bed, and again, with grazing or perhaps root competition can reduce the Syringodium.
If I may, yeah, I agree with that. Without some disturbance, we've actually seen the same thing with Syringodium filiforme, where if you have the shoot density high enough, it doesn't seem like invasive can move into there. And again, sorry, I can show you a quick image of that if you'd like, but yeah, we've seen the same thing. If it's really thick, it seems like that is the best defense against the invasive.
I'll ask a question. Does anybody have any information about sort of the reverse of that, and that Thalassia going in and outcompeting Halophila, because it could shade it out?
Not that I'm aware of, or at least we don't.
Yeah, I'm not aware of that either. So.
We have another question. This is from Ashley Rufo. Has anyone observed conch interaction with Halophila stipulaceae and invertebrate epiphyte community versus native seagrass beds? My apologies.
I have not, in Brewers Bay, we have a lot of, sorry, my phone keeps ringing. In Brewers Bay, we have a lot of work with students, and we don't see conch in the Halophila beds nearly as much as we see in the Syringodium bed. I guess he's calling.
Do we have any more questions?
I have a question about the genetics. Really very interesting. One genotype dominating and another 16 genotypes. Is there any possibility that these multi-locus genotypes could be result of mutation or somatic mutations? Because there are so many, because you had 200 SNPs, so the probability for mutation in any of these loci is could be a bit higher. Is there any way to calculate that?
Well, we would have to first have some way to actually measure the mutation rate in this particular species. So I haven't found anyone who's done that, because I have thought about that possibility and tried to find out, but there doesn't seem to be something that's been done. So if you look at where mutation rates are for plants in general, it seems unlikely that you'd find mutations at that rate. But it's certainly possible that this species just has an exceptionally high mutation rate. I've heard that hypothesis before. I haven't seen any kind of evidence to back that up. Now that might be another study to put on the list for future work. Somebody to find out what the mutation rate is in this species.
Yes, because there even exists a possibility that the first fragments which arrive, for example, if Granada population is the source population for the Caribbean, maybe there were already mutations within these fragments which later on spread. So it's very difficult to know the time frame on which this mutation could take place, isn't it?
Yes, and that's a good point. We can't narrow it down that much. It doesn't have to be just since 2012. The species was in Grenada in 2002, and perhaps somewhere else that we don't know about even earlier than that.
But yes, thank you. Very interesting.
Brigitta, I was going to say that Kelsey's work, we've been able to get 1,200 high quality SNPs. Kind of looking at this question, is there the potential for really rapid evolution? Again, Alice is totally correct. We need to figure out what that rate would be. I do know that the whole genome for this sequence, for this species, has been sequenced, and it's being annotated now by some folks. And so once that comes out, that might be a way to help us get to that question of what is this rate. It is possible to get additional SNPs to kind of ask that question.
Next we have something from Demian. I believe he would like to share a slide on Halophila and Serengodium. Demian,
yeah, I come back to that question earlier really quickly about the sorry, let me look at on the invasive into a healthy bed. And so so this is the paper I referenced in the talk that you can see. This is from Dominica, where we were there in 2008 and 2013. I want to draw your attention to this part in the middle. So this is basically going from shore out into the sea. So from the right, we're at the coast, and on the shoreline, to the left is out into the water. And we found consistently across the island in all the habitats we looked at, that there was basically a region where we called it the Serengodium filiforme stronghold, where the invasive never made it into that region. And that's when we had benthic cover of Serengodium filiforme of at least 45%. And so it made an environment where we had, you know, it's, again, it would seem like it's healthy enough to keep out the invasive. Again, this is the Steiner and Willette 2015 paper. I don't have the data for Thalassia, but we have seen some evidence of this for Serengodium. Healthy seagrass bed of native, I think this is a little bit of indication that that's really worth our effort to keep them healthy as opposed to trying to remove it afterwards.
Okay.
Right. That is absolutely true. I mean, certainly with the damaged areas like we've had in Brewers Bay due to the storms, and again, we have some anchoring that occurs there. The Halophila is, well, as you saw in that bar graph, is pretty much 95% or more of the seagrass in the bay now.
Thank you. Next, we have two comments. The first being from Crystal. Good day, all. I was snorkeling at Willette Bay Island, BVI, and recorded a juvenile conch among Halophila stipulaceae. Next, we also have Fee Smolders. Hi, Ashley. There has been a report written on this topic. It reported equal abundances of conch in native and invasive meadows. approximate response of fish, conch, and sea turtles to the presence of the invasive seagrass Halophila stipulaceae in Bonaire. Next, we have a question from Joe Kenworthy. Has anyone been probing the Bahamas? Robert, the floor is yours.
Is my mic working? Can you hear me?
Yes, we can.
To my knowledge, I have not heard any reports of it in the Bahamas, and from my work there, I haven't ever found it or seen it there, but of course there's a lot of islands and a lot of seagrass coverage there, but I haven't heard any reports of it there yet.
I believe I've heard of it being in Turks and Caicos, but I'm not sure if that's an official report. That was just a conversation I was having with someone who lives there.
Next, we have a question from Jennifer Schul. Have there been any sightings in the Yucatan?
No sightings in Mexico yet. So neither Quintana Roo State, Yucatan Peninsula. I don't think either in Belize, Panama, Costa Rica. The only place where it's been found on the Latin American mainland of the Caribbean is Venezuela until date, as far as I know.
I have a question, although I think we're getting close to our time limit. Has anyone done any of the calculations on the sort of increase in primary production due to Halophila? I mean, it's going down to, 30 meters or so, and covering areas that were never covered by seagrass before. I'm just wondering if anyone has done that sort of back of the envelope calculation. I guess not.
I'm aware of a couple folks who are doing blue carbon sort of studies. I think it is really Grimes who has, I believe she's presenting a talk at ASLO in Puerto Rico early next year that has some blue carbon data. And then one of my students, Kenneth Frost, has looked at this blue carbon, comparing it between Stipulacea and invasive and then natives, and there's a significant difference between at least the Stipulacea and the invasive seagrass of total carbon that they're putting out. But we have yet to kind of do that, back of the envelope. How much carbon is it sequestering? If that kind of question you're asking.
Okay. Yeah, Dr. Wilson Grimes and their student Amalie Jensen are here at UBI.
We have about approximately 10 minutes left. Is there any more questions?
So see, JB is suggesting it would be worthwhile to reach out to School of Field Studies, Turks and Caicos, to report. That was actually, it was Heidi, I'm blanking on her last name, I believe, who told me that they had seen that there in South Caicos.
Antonio, may I say something?
Floor is yours, Andy.
I wanted to thank everyone who participated in this webinar. As Judd said, we tried to have a workshop which was prevented by the hurricane. and it was not clear how we were going to move forward. This has been, to my mind, a very interesting and worthwhile time. All the presentations were good. The questions have been very interesting. There are at least 2 or 3 lines of inquiry that will produce more information and continued study if they get funded. The route, as Demian pointed out, around Southern Africa, that's quite interesting. What Brigitta was saying about the difference in genotypes from one location to another, that's very interesting. Anyway, I just wanted to say thank you very much. It exceeded all of my expectations.
Yes, mine too, Sandy. This is Paul Jobsis, and I want to tip my hat to you, Sandy, who's been extremely patient with trying to get this done. It was started 2 years ago with some inspiration from you, and so it's great to see that it's finally made it this far. I'll also say that due to some funding from Sea Grant in Puerto Rico, we will have this translated, or at least the slides translated into Spanish, so they will be available. And then we're going to host the entire webinar, including me saying a lot, on a UBI server.
Yes, that is correct. The webinar will be recorded, has been recorded, and will be online. I will be sending out the links for that when it becomes available. Within the next week, you'll also be sending out a survey about your opinions and thoughts on the webinar, and we would love to get your feedback on it. Thank you all once again for coming and participating.
All right. Everyone, I think that's it. Have a good day.
Thank you, everyone, and have a wonderful day.
Yeah, contact Antonio if you have more questions.
Please do.